Welcome to clickmyproject.com. Let me discuss about the project Simpler, Faster, More Accurate Melanocytic Lesion Segmentation through MEDS. Let me discuss about the abstract of the process. Physicians employed to diagnose a melanocytic lesions in order to determine with experimentally proposed principal component analysis. These mathematical features are correlated with these physical characteristics analysis. These features have been improved with the principal component analysis and a support vector machine classifier. This method to identify physical features that experienced plastic surgeons employed to classify lesions into their segmentation. Lesion injury is essential to initiate the correct first treatment. For diagnosis, the treatment is necessary to know the segmentation of the lesion. Correct visual assessment of melanocytic lesions relies highly on specialized dermatological expertise. Epidermal lesions are usually red, dry, and painful. The main problem encountered in the analysis of digital photographs of lesions wounds melanocytic lesions from digital photographs have been published with encouraging results. Brainstorming to identify features in the images correlated to their coordinates in the multidimensional space. In our proposed system, physiophysical test has been implemented initially where R, G and B regions are separated. Lesions are manually segmented to analyze the segmentation of lesions. In this step, similarity matrix will be obtained. Next, PCA is a multivariate analysis method has been implemented to analyze similarity matrices for a graphical representation of the data. If the similarity between two objects is low, then their distance in the map must be high. The three axes that emulate the PCA axis with the physical features that physicians use in order to diagnose the lesions. This paper, a physiophysical testing and principal component analysis have been used to detect physical structures that practiced plastic surgeons employ to organize lesions into their segmentation. Images were backed in pairs to 8 plastic surgeons allied to the lesion unit of Virgin del Reposo Hospital, Seville, Spain. A person whose color precision is under evaluation must distinguish numbers or figures inside some originals elaborated to that tenacity. The training test was the same for all the entities, so all of them will get the same facts about the rank of evaluation. Let me discuss about the flow diagram for our process. First, capture the input image from the dataset folder. Here, we need to take the lesion skin structure from the dataset folder. Next, in the image pre-processing, generally medical images are noisy. Noises in the images reduces the quality of the images. In order to improve the quality of the images, we normally employ some filtering operations. Median filter is used for filtering. The median filter considers each pixel in the image in turn and looks at its nearby neighbors to decide whether or not it is representative of its surroundings. Instead of simply replacing the pixel value with the median of neighboring pixel values, the median is calculated by first sorting all the pixel values from the surrounding neighborhood into the numerical order and then replacing the pixel being considered with the middle pixel value. Next, in the color conversion process, we convert our input image into red, green, blue band, that is red region, green region and blue region. Next, in the principal component analysis are used. PCA is the simplest of the true eigenvector based multivariate analysis. Often its operation can be thought of as revealing the internal structure of the data in a way that best explains the variance in the data. If a multivariate data set is visualized as a set of coordinates in a high dimensional data space. Most informative viewpoint, this is done by using only the first few principal components so that the dimensionality of the transformed data is reduced. PC is closely related to the factor analysis. Factor analysis typically incorporates more domain-specific assumptions about the underlying structure and solves eigenvectors of slightly different matrices. PC is also referred to do canonical correlation analysis. CCA defines coordinate systems that optimally describe the cross covariance between two data sets, while PCA defines a new orthogonal coordinate system that optimally describes variance in the single data set. Such dimensional detection can be very useful step for visualizing the processing high dimensional datasets while still retaining as much as the variance in the dataset as possible. First, two principal components finds two-dimensional plane through the high-dimensional dataset in which the data is most spread out. So, if the data contains clusters, these two may be most spread out and therefore most visible to be plotted out in a two-dimensional diagram. Next, in the grayscale segmentation, thresholding is the simplest method of image segmentation. In the grayscale segmentation, we are used the threshold-based segmentation. From a grayscale image, thresholding can be used to create binary images. The threshold at each pixel dynamically depending on the average gray level in the neighborhood of the pixel. This adaptive threshold technique used to adopt differentiate between foreground and background pixels resulted output with high accuracy rate in different illumination. Finally, we got the resulted with the exactly segmented region. Let me discuss about the running procedure for our process. Select the main.m file in the current folder, like that file, and run for executing this process. This is the main window of the segmentation of melanocytic lesion. By pressing the input image button, we can browse the image from the dataset folder. Here, select any one lesion image from the dataset folder. Here I display the input image in the axis 1 plot. By pressing the noise reduction button, we perform the noise removal operation. Here I display the noise reduction image in the axis 2 plot. By pressing the next button, we can move on to the region segmentation window. By pressing the split RGB region button, we can perform the red region of the image, green region of the image, and blue region of the image. This is the color image segmentation process. By pressing the next button, we can move on to the pitch extraction window. By pressing the PCA button, perform the principal component analysis features. Here, select region for getting eigenvectors. So that we need to select any one region for getting the eigenvalues and eigenvectors. By selecting here, we got the red region has been selected, and here we got the eigenvectors for the red band, and this is the histogram representation for red region. By pressing the next button, we can move on to the segmentation result. This is the final result. Pressing the segment button, we can perform the segmentation operation for based on the eigenvector of red region. This is the binary output, and this is the segmented output with the threshold value of 0.62. An assessment should be objective and non-biased, but in practice to rely on clinical observation and visual inspection of the most practical method. Thank you.